Hi everyone. So in this presentation, we're going to talk about how cells stick together and how they communicate and why that's important to our overall body. So let's just go ahead and get started. So cell to cell adhesion means stick together. So it's kind of like glue that's allowing plasma membranes of individual cells to bind together so that they can actually form tissue or an organ. And one important feature of that is the extracellular matrix, or ECM. We're going to talk about that a lot. So really put it in your brain. That matrix is created by cells secreting substances that are formed together this kind of net or meshwork of fibrous proteins, and they're all kind of combined together in a gel-like substance. All of that is incredibly important, those proteins, every little bit of that extracellular matrix. It's actually providing the nutrients for the cells. It's taking their waste products away, and it's allowing water-soluble substances to go in and out of the cells. So that matrix is important. And there are three main things that kind of create that matrix. The collagen and elastin that give it its structure and allow it to stretch. There's the adhesive glycoproteins, which are allowing the cells to stick together. And there's proteoglycans and hyaluronic acid. And that acid is incredibly important to the creation of things like ATP in the cells or protective mechanisms in the cells. So one condition where that hyaluronic acid is missing or it's out of balance is osteoarthritis. And osteoarthritis happens when we get kind of wear and tear on specific joints. So it ends up, because we don't have that hyaluronic acid the way that we need to in the synovial fluid, the joint itself can't get lubricated, so it kind of is rubbing bone on bone quite a bit, and it degenerates or breaks down the joint. So that ends up causing the pain and the swelling and everything that happens in those patients that have osteoarthritis. So I really want you to understand osteoarthritis, that hyaluronic acid in the synovial fluid is out of balance. The basement membrane is also going to be really, really important, and it's a specialized type of extracellular matrix. It's very thin, but it's also very tough and flexible, and it goes underneath the epithelium of many organs. So like in your skin, you'll have a basement membrane. In your GI tract, there's a basement membrane. Everywhere, pretty much, all of our different organs. And there we refer to it as the basal lamina. So there's one condition where that basement membrane is damaged, and this condition is called Goodpasture syndrome. It's really rare. I'm probably not going to necessarily test you on knowing the patho, but I want you to kind of put the disease in your head as an example of what happens when the basement membrane doesn't work, right? So this is a rare autoimmune disease, and it happens when the body's immune system mistakenly creates antibodies that attack that basement membrane in the kidneys and in the lungs. So it ends up causing glomerular nephritis, which is, I know, a big word, but the nephrons in the kidney actually become very inflamed, and their job is to filter. So they're not able to do their job, and so they end up having proteins and all kinds of stuff spilling out into the urine. And this disease also ends up attacking the ECM in the lungs, so they actually have bleeding in their lungs. Bad disease. Let's hope none of us ever get it. Now, the cell junctions, these are connections between neighboring cells that serve specific functions. They hold the cells together to create a tight seal. They provide strong mechanical attachments, and they also provide a means of sending chemicals back and forth, which serve as signals. They also are going to maintain the polarity, that positive or negative electrical balance of the cells. And the cell junctions can be different types. They can be symmetric or asymmetric. The symmetric ones are your tight junctions, your belt desmosomes, your desmosomes, and your gap junctions. And the asymmetric junctions are known as hemidemosomes. You should hopefully remember this from physiology, but if not, just kind of put it in your brain and let's remember it when we're talking about different diseases that impact the way the junctions are created or made. So Here's a couple of different examples of this. What can happen when cell junctions fail? Pemphigus vulgaris, which you see on the picture here, this is an autoimmune disease that targets those desmosomes, which again are the cell junctions that hold the skin cells together. And the immune system attacks these junctions, and that causes the skin to actually blister and become very fragile. And you can see that here. Another condition is connexin-related disorders. This is a mutation in the connexin proteins, which are needed to form those gap junctions. And this can cause various conditions, including things like hearing loss, skin disorders, and even neuropathies. There's also hereditary diffuse gastric cancer, same kind of thing. The cell junctions have failed in the stomach lining, and it's allowed for ulcerations that ends up causing the tissue to change and cause cancer in the stomach lining. And then there's primary ciliary dyskinesia. This one affects the cell junctions of cilia, which are the little hair-like projections that we need to be able to move mucus and fluid out of our lungs. And there's lots more as well. Those are just a few examples. So the cells communicate in three main ways. They form protein channels, which are our gap junctions, which allow proteins to move from one cell to another cell. And those proteins are going to actually cause things to happen in the nearby cells. So they're very, very important. And also we talked about proteins and why they're going to be needed. Well, we will be talking about this in DNA and RNA, why they're going to be needed for making new cells. Okay, so another way that cells communicate is they have little receptors on them. And I'm holding my hands up like this because that's kind of what they are, is like they put out little docking stations or like little locks for keys to fit into. And those receptors allow proteins or hormones or medications or whatever to come to them and actually change the way the cell processes things inside. So they can, and that's what they're doing here. They're using the receptor proteins inside of the target cell. So a lot of times the receptor causes a change in the protein inside of the cell. And there are specific types of intracellular signaling, including contact dependent, paracrine, hormonal, neurohormonal, and transmitter. Signal transduction is going to involve signals, which are kind of like instructions that are received from chemical messengers outside of the cell. So a chemical comes to the cell and the cell goes, oh, I get the message, and then changes everything inside of the cell, takes it into the cell and says, okay, now you need to follow these instructions. But if the cell does not get the appropriate signal that it should have, then it's going to actually communicate to the cell that it needs to die. And that's actually known as apoptosis, which is programmed cell death or cellular suicide. So a little bit more on the intracellular signaling and the different types. So neurotransmitter signaling, this is where neurons release neurotransmitters into the synaptic cleft, which is the little space between the neurons, and that allows the signal to be transmitted. So for example, we have a neuron that sends a signal to muscle cells, which then tell the muscle cell to contract. Neurohormones or neurohormonal signaling, this is where you have neurohormones that get released by neurosecretory cells into the bloodstream. And those hormones then impact something else in the body, make it do something. So for example, when vasopressin gets released into the blood and then causes vasocells to constrict. Hormonal or endocrine signaling, this is where hormones get released in the bloodstream and then they affect distant target cells. So for example, your pituitary gland can release growth hormone, which then tells your body to grow, especially important during puberty, right? Paracrine signaling is where you have signaling molecules that affect nearby cells. And an example of that is going to be inflammation, which we are going to talk about in depth in Module 2. Contact-dependent signaling is exactly what it sounds like. They have to make contact. So this requires direct physical contact between cells. And one example of that would be like an immune process where there's a cell that's infected with a virus or whatever, and the immune cell has to actually make contact with that cell in order to kill it off. Second messengers, hopefully you remember this from A and P physiology. You have the first messenger that gets to the cell. There's a receptor on the cell which receives the message from the first messenger. That receptor then creates instructions inside of the cell via the second messenger, and that is what's going to impact the biological activity that happens as a result. So some examples of first and second messenger. I wanted to use fight or flight because I think it's going to be really important as we move into inflammation and infection for you to understand that. So think of the first messenger. You see a bear, right? That's scary. Your body is now going to release epinephrine, which is the hormone adrenaline, into your blood. Adrenaline is going to act as your first messenger. It's going to bind to the specific receptors on the surface of target cells like your liver cells. Receptor activation is the next step. That's where that adrenaline or epinephrine is going to bind to the receptor, and it's going to activate the G protein on the inside of the cell membrane, which is then going to activate an enzyme called adenylyl cyclase. So the second messenger is actually that adenylyl cyclase. In the body, it converts ATP or energy into cyclic AMP, CAMP. And that CAMP is then actually going to be the second messenger. The CAMP diffuses through the cytoplasm and it activates a protein kinase A, PKA. So what's the response by the cell? Well, once that activated PKA phosphorylates various proteins inside of the cell, it's going to cause a cascade of events. In the liver cells, that results in the breakdown of glycogen into glucose, which then gets used in the bloodstream to provide a quick source of energy. So pause here. That means that yes, in your fight or flight response, one of the things that happens is your body makes more glucose. So stress increases your glucose, okay? That's important. Put a big star by that, put it in your brain because you're going to need to understand it in so many different processes with your patients. All of that action that's happening is going to help the body respond to that stress by mobilizing energy, increasing the heart rate, preparing your muscles for fight or flight. Cellular metabolism and anabolic or anabolism is going to be an energy using process of metabolism. So the cells are using energy. Catabolism is where they're releasing it. They're breaking it down and releasing energy. Remember that ATP is going to work as an energy transferring molecule. It's providing fuel for your cells. We are going to store energy through carbohydrates, fats, protein molecules, but when it gets broken down or catabolized, it gets made into energy in the form of ATP. And you want to kind of review in your mind the phases of catabolism. We have digestion, glycolysis, oxidation, and that citric acid cycle, which hopefully you remember from physiology or chemistry if you took organic chem. Cellular metabolism, just to trigger your memory, involves oxidative phosphorylation, which is very important to cellular function. So oxidative phosphorylation occurs in the mitochondria, and that is important to create that energy from the carbohydrates, the fats, and the proteins. It's a crucial process in cellular respiration, and it has two main components, the electron transport chain and chemiosmosis. So the electron transport chain of the oxidative phosphorylation is the process that starts with the oxidation of NADH and FADH, which are produced during glycolysis and the citric acid cycle. Now, I don't expect you to memorize them, but I do want you to be familiar enough with them. If you see them on a test, you go, oh yeah, I know what this is talking about, okay? Electrons from the NADH and FADH are going to be transferred through a series of protein complexes that are embedded in the inner mitochondrial membrane. And as these electrons move through the complexes, they're going to release energy, which is used to pump proteins from the mitochondrial matrix into the intermembrane space and creating a proton gradient. That is important. So do put a star by that. These proton pumps, we specifically have them in the stomach as well, where they produce acid. And when you get into pharm, you're going to need to understand what proton pumps do, because there's a whole class of medication called proton pump inhibitors. So this is why I'm making sure you're aware of many of the different things that we're dealing with. So the chemiosmosis in oxidative phosphorylation, this is where it's the next step. So the proton gradient that has changed its polarity now generates a form of potential energy that's known as the proton motive force. The protons flow back into that mitochondrial matrix through the ATP synthase, which is this large enzyme complex. And the flow of the protons through ATP synthase is going to drive the synthesis of ATP from ADP and inorganic phosphate. The energy that gets released during this whole process is the main source of energy for the cell. Oxygen is needed. It acts as that final electron acceptor in the ETC, so it combines electrons and protons to form water. And oxidative phosphorylation is highly effective. It produces about 30 to 36 ATP molecule per glucose molecule compared to just 2 ATP molecules that get produced by glycolysis. So we need both systems. We need that oxidative phosphorylation. So why do we care about it? Well, here's why. There's things in our environment that can significantly negatively impact oxidative phosphorylation. And again, that's the process that cells need to produce ATP. So there are some key ways in which environmental factors disrupt that critical cellular function. When we're exposed to toxins and pollutants, things like air pollutant, water quality, all of that kind of stuff. Cigarette smoke, not just if you're smoking it, but if you're getting it through secondhand smoke, air pollution, exposure to radiation, and not just X-rays, but even things like the sun. Too much exposure to the sun can negatively impact that oxidative phosphorylation and make it so our skin can't create the energy that it needs to create new skin cells. And if you're thinking, oh my God, that's how skin cancer is created from sun exposure, give yourself a gold star. You are right. Nutrient availability, things like fasting or a high-fat diet can actually negatively impact that process of oxidative phosphorylation. And thermal stress, like extreme temperatures, too hot or too cold. So all of these environmental factors really highlight the importance of maintaining a high-quality cellular environment to ensure that the cells get to function the way that they need to so that it doesn't negatively impact our overall health. Okay, that's it for this little presentation. You move on to the next one, and hopefully you have a better understanding of cellular communication.